Bom dia. Primeiramente, eu gostaria de
Good
agradecer à
morning.
comissão
So first of all, I'd just like to thank the organizers,
a Carol,
especially Carol and Daniel, for the opportunity to participate in this conference.
tão importante na
This
área do
conference is
Malaria.
so important for our
Nessa
field.
sessão, nós vamos dar
So in this
continuidade
section,
sobre vetores.
we will continue to discuss vectors and transmission.
Eu estou aqui, com Fábio Gomes,
I have Fabio Gomes here with me,
mediar
who will co-moderate the panel.
para fazer a introdução da primeira palestrante.
So I'll invite him to introduce the first speaker. Good morning, everybody. So to open our session, I have the honor to invite Professor Dr. Silvia Portugal. She's a biologist. She has a doctorate in the Molecular Biology Institute of Lisbon. Her postdoctorate is in an NIH under Peter Crompton's supervision, if I'm not wrong. We had some overlap at that time. And since July 2020, she's been a researcher of Max Planck, where she heads a research group, Malaria Parasite Biology, and among other things, she's been working on investigating the mechanisms of Plasmodium falciparum in seasonal transmission. She's published in a variety of journals, so I'm certain it will be a great presentation. Thank you so much.
Bom dia, muito obrigada.
Good morning, thank you so much, especially to Camila and Daniel. Thank you so much for the invitation. I was persuaded to speak in Portuguese, but I prefer to work in English.
To survive the dry season, and I'll do it in English. But I'll be happy to chat in any language then during the coffee breaks later on. So, okay, this is Bamako, seen from the top of the village of the hill where we have the lab, and you can see that the dry season is very sharply demarcated. It's long and is very different from the wet season. And this makes it very difficult for mostly any organism that wants to survive in this place. So, for instance, frogs, they have a very hard time without water, so they have to change very much, especially their dorsal skin, and they sit and wait for better days. And also trees have to completely change their canopy and their roots in order to minimize the need for water during the times when it's not so much available. And mosquitoes really just try to get the hell out of there. They either estivate and wait for better days, or more commonly, they migrate with wind currents that take them to places where there is water available that they need for their reproduction. So this is what's behind the seasonality of malaria transmission in most of Africa, and in Mali in particular, that is highlighted there in red. That is, I don't know if this is going to work. It will. Perfect. We have this very clearly, sharply demarcated malaria transmission season when we have all the clinical cases occurring here between June and December, and then followed by a 5 to 6 month dry season where we have virtually no clinical cases. As I told you, this follows very closely the presence of mosquitoes in the environment, and the difference is that basically during the wet season, everyone is getting infected, most children is getting sick, but during the dry season, parasites still have to be somewhere. And we and others have shown that close to 25% of children still carry asymptomatic parasitemias. And then a big difference in terms of cycle is that during the wet season, you can have the constant cycle beginning because the three partners of mosquitoes, humans, and parasites are present, while during the dry season, the parasite is virtually stuck in doing these asexual cycles in the blood. So when we started the lab, basically the questions that moved us was trying to understand how can these parasites, how can these children carry these deadly parasites for 6 months and completely unnoticed, and what does the parasite have to do in order to bridge these two wet seasons months apart? So I'll summarize 4.5 years of work into one slide. Basically, after extensive analysis, what we found was mostly behind this big difference is that during the wet season, we have the textbook circulation only of young forms of the parasite. Everything else is bound to the endothelial cells of the vessels and is not seen circulated. And here shown the baby parasites in a thick smear. While during the dry season, parasites are poorly adhesive, they circulate longer, we can detect these older forms of the parasites in circulation, and this helps persistence. What we have shown, both with a model of a spleen and through mathematical modeling, is that by lowering cytoadhesion, parasites get lost every 48 hours without adhering efficiently to the spleen, to the vessels, they get removed by the spleen, and parasitemia is not able to increase, and this helps the parasitemia remain low, the lack of symptoms and the lack of immune responses that could clear the parasites. So, as science goes, you answer some questions and you only get new ones. So basically, after finding this, we wanted to understand how could parasites modulate this circulation time and cytoadhesion to become persistent. And I'll be answering a few of these questions during the talk. And basically, the way that we have approached always the ideas that we have had and hypotheses that we design in the lab is always to collect real-world data from the cohort studies that we try to implement in endemic areas. So the lab, I'm not sure you can see any of that, but the lab basically operates between Berlin and Bamako. We started with the very important collaboration of Pete Crompton and Boubacar Traoré from the NIH and the MRTC in Bamako. Later on, when I joined the Max Planck Society, we were able to establish a partner group funded by the Max Planck that is now led by Moussa Nyangali, that helps us run shop and implement the observational cohort studies that we mostly do now in Torodo, where we follow populations below 18 years of age. We collect both blood specimens and clinical data. We do active and passive surveillance. We provide care 24/7. And of course, we treat all diagnosed malaria cases. So this is an example of a basic setup of what we do. During the dry season is mostly through active surveillance. We call all the people to come monthly or whenever it is to the clinic. We collect blood from everyone. We do the RDTs in the lab to find out who has parasites. And then we do the active surveillance, the passive surveillance, that is mostly how we get our wet season samples. People come to the clinic if they feel sick. And if they have a clinical case, we treat, we collect the blood, and then we do the analysis in the lab. So the first type of question that we tried to address in recent years was, how does the parasite know? Is there a type of a sensing mechanism that helps it define when it's wet and dry season? Or is there a time feature to it, or within hosts dynamics of the different clones that is going to help define who is going to survive until the end of the dry season? So to answer the first question, this is all published, so I'll just skim through it very quickly. We did a bunch of omics to look for differences in the plasma composition. We've seen that throughout the year we don't see any seasonal changes. Only if people get sick, then we start to see some differences, mostly in lipids. So we couldn't find any evidence of a sensing mechanism. We tried to put these plasmas in contact with parasites in culture. It never affected. Also, against a sensing mechanism, we could see that type of parasites that we find in the dry season with longer circulation and troughs seen in the blood, we can also detect in the wet season as long as people are asymptomatic, which doesn't play well with a sensing mechanism. What it could detect, however, is that the parasites that we see surviving until the end of the dry season are not randomly transmitted during the wet season. If they want to have a higher chance of surviving, they will be transmitted later in the wet season. Then it increases their chance of reaching the end of the dry season on the other hand. So this made Manuela very interested in trying to understand what is the clonal dynamics that allows parasites to survive these 6 months. So here I have an example of one person. This person was followed every 2 weeks during the wet season. We started in July, and every time we get a filter paper, and then we do amplicon sequencing for a very polymorphic gene in order to define which parasites we'll have there. So each color is a clone. So you can see this child didn't get sick throughout the wet season. It had 2 peaks of parasitemia, one here and another one there, and then eventually this clone survived or not until the end of the dry season. And what you see in this case is that this pink one actually made it to be potentially transmitted when the season restarts. So we tried to get this from a bunch of people and try to understand what is the dynamics that we can see there. So here, just to show a complex slide, but it's all the 128 individuals there. We have three types of scenarios. In red, dark red, we have the ones that actually have a persistent clone that is going to reach the end of the dry season. And then we have the two other situations where there's clearance, either naturally without any type of treatment, at least provided by us, but we are the only clinic in the village, or following treatment if there was a diagnosis of a malaria case. And now I try to put find some patterns in all this data. The first thing that we ask if there would be any numbers of different clones that would promote more or less persistence until the end of the dry season. Oh, I'm finding typos, I'm sorry. And we don't. Basically, the number of clones that a person has in average throughout the time is not different. But we do see that individuals that clear their infections show more peaks of parasitemia throughout the time that they have parasites than individuals that maintain their infections until the end of the dry season. We've also measured what is the length of each of these parasite clones that the individuals show throughout time. And we see that most clones are short-lived. It doesn't matter if they come from children that are going to keep a persistent clone or children that are going to clear them. Most infections are going to be short. And in fact, when we measure the whole length of the whole clones that we have there, during the wet season, we see that it's only marginally higher, the length of each of the clones that are seen in children that are going to be able to maintain persistent clones. Then I want to introduce these two concepts. One is this clonal turnover that is defined by length of each of the clones that you have, and then appearances of clones throughout time, and also the disappearances that you have. So I'm showing here three examples. One of low clonal turnover. There's actually no clonal turnover. They just have that one clone the whole time. One intermediate one, where you have a lot of appearances, but actually not much disappears. They mostly persist until the end. And then a higher clonal turnover rate, where you see that clones come and then others go and others replace them. And the second concept that I want to introduce is this Shannon evenness, that actually is most of an ecological measure that is used to define how biodiversity in a place is. And basically it's just to know what is the relative abundance of a lineage within a total. So again, three examples. Like a non-diverse situation where all the clones, or there's only one clone present, and then two different Shannons with the similar clonal number. So in both situations, we have a COI, a complexity of infection of 3, but this one is more uneven. There's more of one of the parasites, or one of the blue circles, growing. Well, if they are more or less similarly abundant, we'll have a higher Shannon, and this gives us an idea of how even the populations are in total. So we measure these two features in both persistent individuals maintaining persistent infections and individuals clear them. And we saw that individuals that clear their infections have a higher clonal turnover during the time that they are infected. And we've also seen that individuals with persistent infection are actually much more able to maintain a stable infection. And they keep this high evenness throughout time from the beginning of the transmission season until the end of the following dry season, while the individuals that are going to clear their parasites preceding the event of the clearance, they show a decrease in Shannon evenness. So when we put it all together in a generalized linear model, we actually see that the probability of persistence is increased when you have a high evenness and a low clonal turnover. So our persistent infections, or the circles, all piled there, and it's kind of independent of how many clones you actually have. So then, of course, we're going to try to understand what is behind this. And the one thing that we had were the plasmas from these individuals. So we try to see if there's an immune component that is promoting all this. We use plasma samples collected before the season actually started, so before they went through all these transmission events. And we probe this plasma into a protein microarray with multiple hundreds of falciparum proteins. And what we observe is, in fact, that when children are able to maintain their persistent infections, they respond. They have a higher breadth of response to these peptides, and also the magnitude that they respond with to each of the proteins is also higher. And of course, within this array, we can identify increased recognition of the children with persistent infections two proteins that are known for invasion and cytoadhesion. So, okay, trying to put it all together, we are not entirely sure of what is going on, but we do know that persistent infections will happen in children that have a certain level of immunity, and that they will be able to maintain parasitemias that do not fluctuate much. And they're able also to maintain even infections that different clones do not, let's say, outcompete much of the others. And kind of the idea that we have behind this is that these peaks of infection that potentially change the evenness of the composition within the host will probably promote clearance of some of the clones. So this kind of hints to us that maybe it's easier for a parasite to survive during the dry season when you stop having these incoming clones and potential for increased clonal turnover. And that's something that we are very much actively exploring right now. So this kind of tells us who is that can keep parasites during the dry season, but it actually doesn't tell us much on how they're able to decrease cytoadhesion or circulate longer during the dry season. So I'm just going to show briefly work from Hannah Fleckenstein that Maria also mentioned prior, that was in both my lab and now in hers. And also I'm going to show a bit of data about our search to try to define what are the variant surface antigens that these parasites are putting out on the red cell surface to avoid very efficient cytoadhesion. So just to come back to how cytoadhesion happens, we've all seen the beautiful talk of Maria 2 days ago, but just to highlight that during this 48-hour cycle, the parasite transformed the red blood cell from this biconcave, very smooth, very round cell into this very knobby and very sticky erythrocyte that is going to bind the endothelial cells and avoid splenic clearance. And this is done, of course, by the presence of these various surface antigens, particularly the VAR genes that are going to put out the PFMP1s, that are going to promote this very close connection with the red cell. And of course, these are multi-gene families, so they're going to be monoallelically expressed, and we're very interested in what is that the parasites are expressing at each time. So to go first on the host cell remodeling, Hannah, during the time that she was in the lab, did a lot of electron microscopy trying to define if the parasites actually had the ability to transform the red cell to its full extent. So in Mali, getting parasites from the end of the dry season and clinical cases in the wet season, we put them in culture, we let them grow until they are Schizens, and then we max them out to be able to purify them enough to do some transmission electron microscopy. I'm just showing a couple of examples, but Hannah measured everything, number of knobs, height of knobs, density, and we didn't see any difference. Also on the Mauer's clefts, the parasites, if given the opportunity, they will be able to remodel the cell similarly as a wet season parasites. We've also done, just for fun, the cryotomography showing the beautiful network of Mauer's clefts that can be as extensive in the dry season parasite as in the wet season. So they don't seem to be any affected in their ability to remodel the cell, hinting again that it's probably the VAR genes that are not effectively binding the host cell. So to address these questions, Sukai and Martin, in collaboration with Thomas Lafson in University of Copenhagen, we did the BL alpha tag. We amplified it and sequenced, and then we measured how many and which types of PFMP1s or VAR genes coding for PFMP1s we could detect. And basically, another long story put into graphs. What we see is that parasites during the dry season express less of the available repertoire in terms of genome, and they have fewer VAR genes being expressed in the dry season compared to the wet season. We've also seen a slight decrease of total VAR expression that is independent of the VAR type. This was related, or in reverse direction, with the proportion of antibodies that recognize these VAR genes. So again, individuals that keep parasitemia during the dry season, they have a broader antibody responses that is potentially limiting the repertoire of VARs that a parasite can express. Following this initial study, we're very interested in move away from the DB alpha tag and be able to actually look at full-length VAR genes. So we implemented in the lab the SmartSeq single-cell approach that at the broad level of RNA allows us to identify what are similar cells infected with the same clone from different individuals, and then be able to actually mount the full VAR gene by mapping the different domains to the database that Malaria Gen offers at Sanger. So you've seen that this works. I'm showing here just for the VAR aficionados 4 cells, all of them expressing the same VAR gene. The DBL alpha maps to one VAR of the database, while the CIDR will map to another one, and then we'll be able to fully assemble the full-length VAR. So now Chiara in the lab has a full list of samples that we have maintaining the same clones over time during the dry season, so that we can impose or can define what is the pattern of VAR genes expressed throughout time. I have 2 minutes. That's perfect. So basically, just to finish, I want to tell you, and I'm happy to discuss later, I won't have time to show, that we're very interested in finding out if there is seasonal regulation of VAR genes. We've done a single-cell atlas to be able to project the parasites from the dry and the wet season, where we confirm that, in fact, the parasites in the wet season are younger and the parasites in the dry season are older. We've looked for differential gene expression of these matched same developmental time parasites and are now exploring what are these differential expressed genes. And then just finally to show what Martin has been doing, we want to know if this regulation is again imposed by the length of the infections. So we have 2 approaches where we're getting parasites from CHMI and we culture them in vitro for a long time. And we're also looking in endemic region in Mali for parasites that have been recently transmitted versus parasites that have been there for a very long time. So just to tell you what we are doing now the questions that we want to answer, we always have every year a few Plasmodium malaria parasites that also survive the dry season. We actually see more of them in the dry season than we see in the wet. We want to explore what is their toolkit. We want to know what are the buildup of these antibodies over time that limit the VAR gene expression. And in collaboration with Ilana Lavashina, we want to deconstruct also what is the mosquito component that helps. Mosquito, different species come and go throughout the time, and we want to know how much they help make the reservoir distributed through the new wet season. And this is everyone in the lab, all the team in Mali and the collaborators that helps us, and of course the participants that allow for all of this. And I'm happy to take questions. Thank you.
Então, para dar continuidade, eu tenho o prazer de convidar a professora Maria
Let
Anice
us
Salom.
continue. Please, Maria Anice Salom. She's a professor in epidemiology and public health and Culicid taxonomy in the Public Health College of the University of São Paulo. She studied in the University of São Paulo and obtained her doctorate in the same university in Culic systematics, with a postdoctorate in Anopheles in the Natural History Museum of Washington, and acted as a visiting professor in the unit of biosystematics in the Switzerland laboratory. Returning to Brazil, she coordinated various projects in biology and Culicide systematic, with emphasis in anopheline. She expanded her research in field research in the Amazon River Basin, especially concentrating on the ecology and impact of climate change and on the increased risk of infection by Plasmodium. Throughout her career, she built strong relationships with other institutions, and she has a wide network of collaborators, both national and international. And I had the pleasure to start this collaboration with her last year, and I consider Professor Salom really an exemplary researcher in the field of entomology.
Desculpa. Eu inicialmente dou bom dia a todos e todas que estão
Good
presentes.
morning, everyone.
E agradeço imensamente a Ana Carolina e ao
Thank you so much to Ana Carolina and Daniel Pegri.
Bárbara pelo
Daniel Barbary, yes. Thank you so much for the invitation to take part in this event, and thank you also to the organizers.
É uma honra estar aqui.
It is an honor to be here and to be able to talk a little bit about my research.
Bom, eu vou falar sobre
I will be talking about recent results.
Não, instalou. Ah, tá. Desculpa, eu estava no botão errado.
I was pressing the wrong button,
Não sou
forgive
muito
me.
amiga de botões.
I'm not technological.
Só para
Anyway,
colocar dentro de contexto que eu vou falar.
just to contextualize, Anopheles delegia is the dominating factor for Plasmodium vivax in the Amazon region, as well as in the Guiana Shield. The dynamics of the malaria transmission must be understood within an ecological context because it depends on a variety of factors. Among these are the biotic factors, the abiotic factors, also factors related to human ecology, to vectors, as well as to the interaction of the parasites with the human population, with the vector population. And this process is mediated by an environment which is susceptible to the contact of these populations. Well, this is the work that was developed by Andrea Costa. It was published in 2026 by Instituto Oswaldo Cruz. It is an open access work. There were 384 collections in 70 paradomestic sites, which were independent, in 4 distinct ecological zones of the Amazon rainforest. These are these yellow points, which I won't even try to point to. These yellow squares. These collections were conducted in the municipality of Atalaia do Norte, Benjamin Constant, Barcelos, Coarim. In Coarim was south of the Amazon River, all of that in the state of Amazonas, as well as in Calçoene in the state of Amapá. This is a diagram to explain how the collection environments were planned. We attempted to do an ecological sampling as well for these samples to be independent. 1 domestic site was selected. Each domestic site must be around 100 meters from a wet region, which could be an appropriate environment for a mosquito. About 100 meters. Of course, there was some variation. The collections were made from 6 p.m. to midnight. The mosquitoes were separated by hour. And throughout the period of collection, we took a small meteorological station out to the field and set out some important parameters for the climate, including relative humidity. We got around 14,000 females. This is a diagram to display the time distribution of Anopheles darlingi, but also in the context of the human bite rate, the HBR, and the total bite rate by each hour. The difference between these two is that the HBR is utilized by collector and the TBR is utilized by 2 collectors. The collections were always made by 2 collectors. And what were we able to observe? That there was a decline in the human bite rate of about 154 bites per hour, and in the TBR we saw about 340 bites, a decline of about 340 bites per hour, all that throughout time. a significant decline after 9 p.m.
E as correlações foram
And the
significativas,
correlations were
elevadas,
quite significant.
né? Aqui esse scar plot para mostrar o impacto
This is
da
car plot
temperatura
to display the impact of temperature
da BR, ou seja,
in relation to the HBR,
em uma pessoa.
so
Então, a gente
the
observa
human
que
bite
a
rate in one
temperatura
person.
apresenta uma
We observe that the
associação
temperature is positively correlated to the human bite rate,
cada aumento de
and
Celsius,
with every
observamos
degree Celsius increased,
de
we see an addition of 7
por pessoa.
bites per person per hour.
Essa correlação é baixa,
This is a low correlation.
ela,
However,
de qualquer maneira, ela existe e é importante.
it is
sendo
existing, it is there, although it is not high.
Em relação à umidade do ar, a umidade relativa do
In
ar,
terms of the relative humidity of the air, there was a negative correlation with the human bite rate per collector.
também tem uma associação, não é elevada, mas
Also not a high correlation, but a significant one nonetheless.
1% de
And with
aumento da
each
umidade
percent of increase in the relative humidity of the air, we see an increase of 2.5 bites
de aumento de 2,5 picadas por hora por coletor.
per hour per collector,
Então, a cada aumento de 1% da umidade.
with each percent increased in the humidity.
Em relação à cobertura do solo,
In terms of the soil
a
coverage,
taxa de picadas,
as well as the human bite rate,
Anopheles darlingi, ele apresenta
Amorpholis darlingi and its presence in the collections,
aparentemente,
it
ela está muito
was
associada
apparently correlated, and this has been demonstrated by a variety of papers. It was more prevalent in deforested environments.
Então, tem uma abundância maior de
So
darlingi.
in these environments, there's a higher abundance of darlingi, and therefore
Os autores observaram que também
there is a higher human bite rate.
né? A mesma coisa foi observada aqui.
The same thing was observed here.
Foi analisado dois buffers.
In this work, we analyzed 2 buffers, one with a radius of 250 meters, starting from the domicile site,
Círculo imaginário com raio de
and an imaginary circle of 500 meters,
do domicílio.
starting from the domestic site.
Foi observado, né? Que quando analisa o
We
círculo
observed that when we analyze the radius of 250 meters, this area determined by this radius, it is very easy to capture environmental alterations as well as changes in temperature and humidity in these environments. But in order for us to actually understand what takes place with the mosquito population that reaches this domestic household to feed on human blood, it is best to observe the buffer of 500 meters, starting from the household, because you're able to, through this analysis, capture what takes place in the surroundings of that household, not just in the household itself, in a larger area. And we observed that the increase in the human bite rate by Anopheles darlingi can be explained by the vegetation, the floodable vegetation in that region, in addition by humidity, temperature, and by the composition of the surrounding forest within that 500-meter buffer. However, for the vegetation and the floodable region, as these factors are adjusted by the humidity and temperature, the contribution of these two variables for the oscillation of the human bite rate are not as representative, which they would otherwise have without the adjustment of humidity and temperature. This is another paper published by J. Kennedy, postdoctorate in Harvard, study under Daniel Dembski. I was a part of this paper. Some of these samples of this paper derived from the previous paper that I mentioned. In this project, they analyzed 1,024 complete genomes, 1,094, forgive me, from 16 locations in Latin America across 6 countries. It was possible to analyze 1,024 mosquitoes for the calculation of the allele frequencies, but a lower number of mosquitoes was utilized for the core genetic analysis because this depended on the coverage of sequencing, the coverage of the sequencing done within the platform. So the results, or some of the results. In these populations, and regardless of the country, they have a high diversity of nucleotides. Heterozygosity exceeds 2% in most individuals. When only the X chromosome is analyzed, so this diversity is even higher. It can reach up to 13.2%, much higher than it was already observed in Anopheles gumbi and other gummy species that occur in Africa. Anopheles dorsalengi also exhibits a higher population. It's a long-term stable population, and there was a bottleneck. There was a bottleneck in the population from about 4,000 to 20,000 years ago after the last glacial maximum. This is associated with refuge areas, so vegetational areas that were distributed across Brazil and the Amazon region. The genetic diversity can also be visualized by the number of inversions in the chromosomes. Can more easily observe that here. Some inversions are segregating, and they vary from 13 to 19 megabytes. And it is possible that these inversions are associated to the process of adaptation of the species, and therefore it may contribute to the high genetic diversity of these populations. The geographic structure observed 3 primary clusters. First, Amazon Atlantic, this area here, the Orinoco cluster, those blue circles, which include Venezuela and Colombia, and Chocó, also in Colombia, which is closer to Central America, south of Panama. So what could be associated to the formation of these clusters? The Andes cluster, especially in the Chocó cluster, the geographical barrier is the Andes, because this is west of the Andes. So the Andes is a huge barrier that separates this population. In other places like in Orinoco and Atlantic Amazon, the Orinoco River, or rather the Orinoco watershed, is the barrier that separates the populations that are evolving in Venezuela and Colombia from the populations that are being developed in the Amazon watershed. In terms of the response of Anopheles dorsalengi to insecticides, what did we observe? These genes, which of course are undergoing selection, there's a very strong selection sign being observed there in chromosome 2RL, and the group of genes which is P450, and the most important genes of this group are these CYP6AA1 and CYP6M2, which present the highest signaling of selection due to exposure to insecticides. We also observed the KEEP1 gene, which is also associated to or presents signs of positive selection. And these groups of genes, both P450s and KEAP1, are associated to a mechanism of metabolic resistance. This has favored Anopheles darlingii. So what do insecticides have to do with agriculture? Is there a relationship, or is this resistance being bolstered by control programs? It is actually both. In the Loreto region, as well as in the Guinea region in Colombia, we have observed that this resistance is associated to the agricultural utilization of insecticides, especially in mosaic plantations. They utilize that for palm oil and other cultivations, and these are staggered. When insecticides are utilized in monocultures, we see soil contamination, and the insecticide percolates through the soil and reaches water bodies, which leads to a natural selection process of Anopheles, in this case Anopheles darlingii, due to the contact with the immature stages of life and the presence of these insecticides in water bodies. In terms of KEAP1, this was found in the Barcelos population. So a different process may be ongoing there because it does not have that type of cultivation. But an important point here is that this process of resistance and genes, metabolic resistance, it is evolving in parallel in different regions, in different populations of Anopheles darlingii, and it could involve different genes. So this is quite complex, and we need to be attentive towards this evolutional process for metabolic resistance, which is different from the target genes, KDR, ASI, and other genes, in which we did not find any evidence of resistance correlated to these genes.
This also leads us to a need to control the vectors for the elimination of malaria. We have to think about the vector and the environment where this vector transmits, and also the problem of the genetic evolution that is happening actively in Anopheles darlingii. We should also think that it is not enough for the ministry to work on the control of the mosquitoes and the control of malaria. We need to come up with projects with the Ministry of Agriculture and with the Ministry of the Environment, because actions of these other ministries contribute to the selection of resistance in populations of Anopheles darlingii. These projects were funded by NIH and FAPESP, and I thank you all very much for your attention.
I will present in Portuguese and then right? Ok, então, dando prosseguimento, a gente tem a alegria
So
de...
moving on, we have the joy now to have the presentation of Dr. Joel Vega Rodriguez. Joel is a doctor in molecular biology at the University of Puerto Rico, and he had a long period of postdoc in Johns Hopkins under the advising of Professor Marcelo Jacobs Morena. Since 2018, he has led a research group at NIH, the Laboratory for Malaria and Vector Research. And in his line of research and postdoc, and now in his own laboratory, he focuses on bottlenecks of development of Plasmodium of the Anopheles mosquito that has to do with transgenics and vaccines of transmission blockage. Joel, it's a great joy to have you here. Thank you very much. Muchas gracias.
Thank you. Thank you for the introduction, and thank you to Ana, Beto, and to Daniel for the invitation. Ana made me promise 2 years ago when we were in Berlin that I was going to come to this meeting, and even with everything that happened in the past year and the past few years, I was able to make it, and no regrets. You have done an amazing job in organizing this conference. It has been very rewarding, scientifically, and also the social part as well. So my laboratory at NIH works in understanding host-parasite vector interactions that are taking place inside the mosquito. This is mostly happening in the midgut, in the transition of the sporozoite delivery into the skin. But now we found a new area where these interactions are happening, which is in the hemolymph of the mosquito. And the title is Host Manipulation by the Malaria Parasite. Because we see the parasite as this master conductor of two hosts, the humans and the mosquito. And the previous presenters nicely showed how the malaria parasite modulates the human physiology many times to enhance its development and then transmission. This has been very well characterized in many aspects, many biological pathways in the humans. Now we are gaining more and more knowledge in the mosquito, but we are still quite far behind in comparison to the human. So this is the focus of my laboratory. Here we have the development of the malaria in the mosquito. I'm not going to go into detail in it, but I want to mention a few things. Different from the development in the human, the parasite in the mosquito is completely extracellular. And it develops first in the blood meal. Our laboratory has been working for quite a while now on how the parasite takes advantage of blood factors and the saliva of the mosquito to complete development of the parasite in the blood bolus. Then the other portion is in the hemolymph. In both of these environments, the parasite is directly in contact with factors, detrimental factors that are coming from both of these hosts, but also factors that benefit parasite development. We're going to talk about some of those today. Something else I want to mention is that the parasite breaches the barrier of the peritrophic matrix. This is the matrix that protects the epithelium and the rest of the body of the mosquito from all the harmful molecules that are going to come from the blood digestion and the blood meal. So this project started as a fishing expedition. Fishing expeditions sometimes are good, especially if you pay attention to the interesting findings. All we wanted to know was, how is the parasite changing the proteome, the humoral proteome of the mosquito, and how are those changes facilitating parasite development? So we decided to do this by analyzing the hemolymph and the salivary gland and saliva proteomes of infected and non-infected mosquitoes. So today I will present one project from each of those proteomes that have come. So let's start first with the hemolymph proteome. Both proteomes were started by Tiago Luis Alves Silva, staff scientist in my lab from Rio de Janeiro. So for the hemolymph proteome, the study design was we had mice infected with Plasmodium berghei or uninfected. We have mosquitoes bite those mice. And then we did perfusion at three different time points: 30 hours when the oocyst is forming, 10 days when the oocyst is developing. It's supposed to be a quiet time, but it's not. And then 19 days when the sporozoites are released into the hemolymph. And the way we collect the hemolymph is, there is a needle here, you probably don't see it, with buffer. We inject that buffer into the thorax of the mosquito, and then we make an incision in the abdomen, very small, so we collect that hemolymph that is coming out. And as you can see, we don't pierce the midgut in that process. So we observe many changes in the mosquito proteins across the 3 time points. But I will not be talking today about mosquito proteins. I will be talking about mouse proteins, because at 30 hours, at the 30-hour point, we saw something very interesting. We observed a collection of proteins from the mouse that were crossing or detected into the hemolymph of the mosquitoes. They were enriched in infected mosquitoes. At first, we thought this was a contamination, or that the parasite is making a hole, so this is just leakage that is happening into the hemolymph. But if you pay attention to the proteins that are there, then you will realize this might not be the case of a leakage. The reason is, yes, you have hemoglobin, albumin, which are some of the most abundant proteins, but you have proteins here that are not the most abundant in plasma or in serum, like, for example, hemopexin. The second factor that was very interesting is that there is a group of proteins here that are involved in heme and iron metabolism. You have hemoglobins that contains heme. You have albumin that binds heme, hemopexin that binds heme, and then other proteins that are involved in transferrin that binds iron, and proteins that are involved in regulation of oxidative stress, control of oxidative stress. Once we saw this, we started thinking, okay, maybe there is something here that could be important. And if you think about the biology of what's happening in the mosquito midgut when it ingests a blood meal, you would realize that maybe these proteins are having a role in the mosquito, in the hemolymph of the mosquito. So when the mosquito takes a blood meal, the main role of that blood meal is to provide amino acids for egg development. Most of them come from hemoglobin. Hemoglobin is 90% of the blood meal dry mass. That hemoglobin is going to release tons of heme. And as you know, free heme and the iron that it contains is highly toxic for organisms. It can cause permeabilization of membranes, oxidative stress, cellular dysfunction, DNA damage, and inflammation. So the mosquito has some mechanisms to counteract this free heme. The peristrophic matrix bind heme and induce the aggregation of this heme to neutralize it. The mosquito also has antioxidant enzymes as well as the parasite. But the question is this enough to protect the mosquito from heme, from free heme? So we decided to study one of the proteins that we found in this proteome, hemopexin. And this is work done by Francis Saraiva, also from Rio de Janeiro, postdoc in the lab. Why hemopexin? Hemopexin is the protein in our blood that protects us from free heme when we have hemolysis, hemolytic events. It has a high affinity for heme, 1 picomolar, and it prevents heme pro-oxidant and pro-inflammatory effects. And you can see here the pathway for heme. Once heme is bound to hemopexin, it's removed from the circulation by the liver. Okay? So here is then the detection of hemopexin in the hemolymph by Western blot. We wanted to confirm whether we could see it. Here is at 30 hours on infected and infected mosquitoes. You can see there is more hemopexin in the hemolymph of these mosquitoes. Very interesting, we still see hemopexin 10 days after the infection. So that hemopexin remains intact in the hemolymph for quite a long time. And the detection of hemopexin in the hemolymph is dependent on parasite invasion. So we use a knockout for MEOP. This is a protein in the ookinete that is needed to enter the midgut epithelium to invade it. The ookinetes are formed. They touch the midgut epithelium, but they cannot enter it. And in those mosquitoes that fed with those parasites, you can see that there is no enrichment of hemopexin. So you need the invasion point, or the invasion of the midgut to have that enrichment. Where is hemopexin in these mosquitoes? So here you have an immunofluorescence of an ookinete invading a midgut cells. You guys are in the hemolymph, the ookinete is coming towards you. Hemopexin is in green. So what you see is that the invaded cell contains the most hemopexin. However, what you will see is that the non-invaded tissue, the non-invaded cell also start uptaking hemopexin. If you do the same with a non-infected blood meal, it's almost completely black. You see some dots. There is some uptake, but not at the same level. And you see this interesting accumulation around the nucleus, which potentially is the localization also of hemoxygenase in the mosquito, which will be the protein that then degrades heme. So this is then pointing us that potentially once the parasite breaches that barrier and enters the midgut epithelium, the epithelium is reacting to bring hemopexin in, potentially inside the cell as well as in the hemolymph. Okay, so what happened now? What is the role of hemopexin for the mosquito? There is a hemopexin knockout mouse model developed by Emanuela Tolosano from Torino University in Italy. We got those mice from her. So we decided to feed mosquitoes on hemopexin wild type or hemopexin knockout mice. In the first experiment, we did the feeding with dextran because we wanted to see what happens to the midgut barriers in the absence of hemopexin. And as you can see, in the absence of hemopexin, that dextrans doesn't remain only on the blood bolus. You can see then that it enters the midgut epithelium, while you don't see that uptake in the midgut epithelial cells in the mosquitoes that fed on wild-type mice. Then this is suggesting that the peritrophic matrix might be compromised, and that is the case. We did WGA staining, and as you can see, the peritrophic matrix has been disrupted. We think that the peritrophic matrix indeed forms, but then the amount of heme that is and the reaction of heme with the peritrophic matrix then is disintegrated in it. And then the next question is, the peritrophic matrix barrier is damaged. What happens to the midgut epithelial cells? So we look at damage in the midgut epithelium using a transgenic mosquito line from Anopheles stephensii, in which the stem cells of the midgut express the marker TD tomato. This is a line that Carolina Varillas has. And when there is damage in the midgut, these red cells that you can see on the top expand, they grow and they divide. And what you can see here is that the mosquitoes that feed on hemopexin knockout mouse has much more damage to the midgut epithelium. So what we see in the absence of hemopexin, there is a disruption of the midgut barrier. So what happens in the hemolymph? Does this damage reaches the hemolymph? So we isolated hemolymph and we measured hydrogen peroxide levels. As you can see in the mosquitoes that fed on wild-type mice, this was already known. Infection increases hydrogen peroxide. But when you have then the feeding on hemopexin knockouts, whether it's uninfected or infected, you have more oxidative stress in those mosquitoes, or hydrogen peroxide. We also measure hemolymph lipid peroxidation, lipid peroxidation in the hemolymph. And as you can see here, there is an increase that is induced by parasite infection. But when the mosquito feeds on an uninfected mouse, you can see there is much more peroxidation of the hemolymph. Infection increases, but not that much. And something very cool here is we were able to develop a recombinant hemopexin that has 2 point mutations in the amino acids that coordinate the binding to heme. So this is going to tell us whether the binding of heme is what is causing, protecting or not the mosquito. And that's indeed what happened. When we inject in the mice with wild-type recombinant hemopexin, the levels of lipid peroxidations are down, and then we cannot rescue it with the hemopexin mutant. So this is answering that question. Hemopexin is protecting the mosquito by binding heme. That's the actual mechanism. We look at mosquito survival. If you feed the mosquito repeatedly, 3 times in this case, in hemopexin knockout mice, you can see there is a dramatic reduction in survival. And then the number of eggs that are laid by these mosquitoes is reduced. The number of hatching from those eggs are reduced too. So this hemopexin is a factor that helps with vector competence, maintains vector competence. What happens with the parasite, of course? So we did the experiment. This is with Plasmodium berghei. You can see the comparison of all these numbers. Mosquitoes that fed on wild type or hemopexin knockout mice, there's a reduction here. If you inject hemopexin, so this is the same mouse, you feed a group of mosquitoes first, then you inject hemopexin, feed another group of mosquitoes, so you rescue the infection. And then if we do the same experiments with the mutated hemopexin, we cannot rescue that infection from the hemopexin knockout mice. We did the same experiment with falciparum by isolating the serum from these mice and then doing the feeding with falciparum, we see the same result. But we saw hemopexin in the hemolymph, so in the regional results. So we We know that this effect we're seeing here, part of it is from killing of the parasite in the blood bolus, because there's so much heme. So we wanted to know, is hemopexin helping the parasite in the hemolymph? So we did a different design here. We infected mosquitoes from an infected hemopexin knockout mouse. There is no hemopexin in the system. We waited 2 days for the oocyst to form, and then we injected hemopexin in the hemolymph of the mosquito, and then we counted oocysts later. What we observed is that hemopexin maintains the infection in the mosquito. If we inject PBS or heat-inactivated hemopexin, the numbers are lower. And if we do the same experiment with the mutated hemopexin, we can see also that the wild-type hemopexin also enhances the infection. So hemopexin retains its role of binding heme in the hemolymph of the mosquito, and it protects the parasite beyond the blood bolus. So the take-home message here is that these proteins that are crossing into the hemolymph of the mosquito, they do retain their function. And in the case of hemopexin, what we think is happening is that the mosquito has evolved for when there is a disruption of the peritrophic matrix, and now the rest of the mosquito is in contact with factors from the human, especially heme. The mosquito has evolved to bring this hemopexin in and other molecules to then be able to handle the toxic effects of heme. Second project, salivary gland. This was done by Tiago Silva and in collaboration with Fotini, Sinis, and Sachi Kanatani from Hopkins University. And this is a beautiful salivary gland of distal lateral lobe from the salivary gland that Tiago imaged. And so a few points here about salivary gland. This is how the salivary gland looks like. There are 2 per mosquitoes. This is an image from Paulo Pimenta from a long time ago. He described the invasion of the sporozoites in the salivary gland. You see big allineation and Aedes aegypti, and it's very similar in the other mosquitoes and parasites. So what you have is that the parasite gets in contact with the cell. It forms a tight junction similar to the invasion of the red blood cells, and it goes in a membrane. And it has to do that in the salivary gland, otherwise it will kill the salivary gland cell, it will destroy the salivary glands. However, you can have hundreds of sporozoites crossing the same cell, and these cells are not very big. These are factories of saliva production. They have very little cytoplasm, and this huge cavity where they secrete the salivary proteins and where the parasite then is allocated after the invasion. In 1984, Rossignol, José Ribeiro, and Spielman, they showed that the sporozoites increase the mosquito probing time. The probing time is when the mosquito is looking for that blood vessel, okay? And during that time, the mosquito spits. And the parasite reduces apirase activity. So at that time, they call it a biochemical lesion in specific regions of the mosquito salivary gland. In those regions that the parasite invades, which are the distal lateral lobes, these portions here. Okay, remember that study and what I just mentioned about the biochemical lesion. I will just show data from the salivary gland proteome and the biology that we are pursuing here. What happens with the classical saliva proteins in infected versus non-infected? They did not have a significant change, only 2 proteins, D7R3 and a nucleotidase. However, there was a trend in all those proteins to have a reduction in the amount of protein, and that happens also with the saliva protein. What was interesting is when we look at the differentially expressed proteins for all the others, this is just a set of those proteins, we found something very interesting here. Again, the weird result is what is giving us the interesting biology. We found a bunch of hemolymph proteins that were now detected in the salivary gland of the mosquito, including some of the immune genes, the CLIP genes, TEP15, PPO6 is supposed to be a hemocyte-specific protein, and other proteins like lipofourin, which is right there, which is produced by the fat body. Okay, so then the question is, how is this real? Is this an artifact? So we look at a couple of these proteins by immunofluorescence. Here you have Plasmodium berghei in the salivary gland in green. PPO6, like I said, is a hemocyte-specific protein. And you can see that PPO6 is accumulating right where the parasite is. It's basically attached to the parasite. You can see some hemocytes attached to these salivary glands as well. Lipofourin, very abundant protein in the hemolymph. We also see that it's only present in invaded glands, but it's not located with the parasite, and it's not present in uninfected. So these proteins are present, they're there. So how are they getting there? Three potential explanations. First one, is the parasite carrying these proteins? Yes and no. Some of the immune proteins are coming maybe attached to the parasite, but lipophorin, which is highly abundant, is not attached to the parasite. It's not coming with the parasite, so it will not explain how all of these proteins are coming in. Is the salivary gland expressing these proteins? No. We did qPCR, we did RNA fish. We don't see expression of mRNA of these proteins in the salivary gland. Is there damage by the parasite? And this was at the beginning a little bit, we were not sure because about this, Oliver Silvich just published that the parasite doesn't kill the cell, doesn't damage the cell because it goes in with a tight junction and a vacuum. But we still pursue this, and I'm running out of time, but I'm going to try to go through this very quickly. So here we combine the salivary gland proteome and the hemolymph proteome from the 19 days post-infection. What we observe is between these two proteomes that the most abundant hemolymph proteins are coming into the salivary gland, and the most abundant saliva proteins are coming out of the salivary gland. So this is not specific, like in the midgut, this is whoever is most abundant is coming in, whoever is most abundant also is coming out. Okay, there is a disruption of the gland, question. We did a dextran diffusion assay. In the moment of invasion of the salivary gland, we injected dextran into the hemolymph, dissected the gland, and what we observe is that the invasion of the gland, the invaded glands absorb much more dextran than the non-invaded, and we quantify this. And then there was something very peculiar that we noticed with electron microscopy. So this is the uninfected gland. This is 3 cells. This is the salivary gland cavity, and you can see here the membrane-membrane contact of this non-infected gland. And you can see here what happens with an infected gland. The membranes are separated. There's a disruption of that membrane-membrane contact. And something very interesting that we noticed, this has been in the literature for very long. Paulo, it's in some of your images too. This is how the saliva looks in an uninfected mosquito or an uninfected cavity. This is how the saliva looks in an infected cavity. And we are calling this clotting of saliva. We know that hemolymph is coming in. The hemolymph of the mosquito is like our blood. It will clot whenever it's outside of the hemocoel because it will prevent then bleeding. So we confirm this with images from Freddie and from Isabel. And Sabrina Absalon also confirmed with expansion microscopy, just to make sure it was not an artifact of electron microscopy. You can see here even an invaded gland, and in the same gland that has been invaded, there is a cavity that has not been invaded. You can see the saliva looks normal. And we use the RON11 knockdown parasites. These parasites cannot invade the salivary gland. Actually, they try to go in between the cells, but they cannot enter the cavity. And the saliva composition looks the same as the uninfected salivary gland. Yes, I'm gonna finish here. So I'm gonna take you back. I'm gonna skip the model because I think it's obvious what we're showing here. But I'm gonna take you back to the paper from that I mentioned before. This is in their discussion. Thus, a possible strategy for salivarian parasites might be to enhance transmission by damaging the salivary gland. This video from Vanderberg shows you how the parasite is transmitted. When the mosquito is probing, it spits saliva, and with it is coming those green fluorescent sporozoites. So a mosquito that has a damaged salivary gland with clotted saliva full of parasites will have to probe longer and harder, and that at the end will only benefit the parasite, because more parasites will be delivered into the skin. Okay, so that's our hypothesis right now. Okay, and then with that, I want to thank everybody, and I already mentioned the main people from the lab, and there's a lot of collaborators that contributed to this work. Thank you.
Thank you so much. I would like to invite all the speakers to enjoy the table, and we'll have 10 minutes to questions.
Hi everyone, thanks for the talks. So I have a question for Sylvia. So you mentioned that 25% of the kids have, during the dry season, have low adhesion phenotypes, right? And then the parasites might be cleared in the spleen, right? That's what you said. And I would like to retrieve, I think, in that cow paper, they show that those falciparum parasites in the spleen are actually culturable, which means that they are alive. So my question is there a possibility that those kids actually have transmissible parasites in the spleen that are actually like perpetuating transmission during the wet season?
Thank you for the question. I think it's possible. I think that our patients are probably where Matthias put his diagram of like the very severe, the very chronic. Our patients are short chronic or light clinical malaria, so I think it's possible. I also think it's not needed. All these patients have a huge reservoir producing gametocytes also in the periphery. So although I think it's possible, I'm not sure it's a huge contribution to the system of perpetuating transmission in this particular system.
Thank you. Can I ask another question?
Yes, please.
So my other question is for Joel. So you're saying that there is a lot of oxidative stress in the mosquito that is actually like controlled by host mediators. But correct me if I'm wrong. You have a paper recently with Rosado Quinones on GST inhibitors on a parasite. So and you show that actually GST inhibitors might be important to as a transmission blocking drug eventually. So my question is, how much room is there for actually like parasite machinery to control oxidative stress in the mosquito?
So I think there is a lot, because remember what we are creating in the hemopexin knockout is an artificial environment, right? Where we are removing, to me, that will be the main molecule that will be chelating him, then together with the peritrophic matrix. That's independent of then the parasite mechanisms to deal with the remnant oxidative stress when all those systems are in place. So I think this is a combination. Remember that the mosquito has the major bottleneck for transmission. Actually, people talk about the bottleneck in the ookinete stage, but the majority of the killing is in the gametes, and then the zygote and the ookinete in the midgut. So what I think is happening is a combination of factors that helps reduce that damage of oxidative stress, hemopexin, the peritrophic matrix, and then the parasite has its own mechanism, antioxidants as well. So yes, remember the knockout is artificial. It's just showing that that molecule has a major role in maintaining that environment.
Thank you.
Also for Joe, very nice. I don't know if I understood correctly, but the hemopexin goes to the hemolymph passively because of the damage when the parasite invades, or there is any kind of active mechanism that brings the protein in?
We are working on the mechanism now for like the continuation of this project. We don't think it's passive. The reason is, if it was a passive transfer or like a passive going through a hole, you will expect other abundant proteins from the serum to be crossing. Plasminogen, the protein that we have been working for so long now, is one of the top 20 most abundant proteins. It's not detected in the hemolymph. So we really think that it's being brought in by the midgut actively. Whether this is something that is hemopexin specific, or these are vesicles that are carrying other proteins as well, then that's the next step for it.
Very interesting. And well, the mosquitoes are getting blood meals independent of the parasite for hundreds of thousands of years. So why do you think they need the host protein to protect them? They cannot produce their own antioxidant system.
Oh, the mosquito will be like a parasite as well, right? They are taking advantage that we have our own mechanism to protect us. And in the case of a disruption, the parasite is not the only one that will disrupt that peritrophic matrix, right? If there is an imbalance in the microbiome, that peritrophic matrix doesn't form, or it forms weakly. So there are other events that could be happening to mosquitoes in nature that will force them to evolve mechanisms for when that peritrophic matrix fails, they still need a mechanism to protect themselves. And one of those could be then proteins from from the host. And we detect hemopexin in the hemolymph of non-infected mosquitoes that fed on non-infected mice. So there is some import in. It's just that we see a huge increase in the staining in the cells when the parasite invaded the midgut.
I have a question for Sylvia. So you showed, you analyzed the VAR gene repertoires of the patients. Did you do that also per patient over time? And so you showed also that clonality changed, for example, when you had a peak in parasitemia. Is that maybe because the VAR gene repertoires changed then?
So in that particular study with the DBL alpha, we had 2 out of the 12 of 13 patients that we did that we had pair samples beginning and end of the dry season. I think in both of them we had common clones in the beginning and end. We also had non-common clones. So all of it points that there is switching, but it's very hard to tell unless we are able to say which clone is producing which VAR, for which I think the single cell will hopefully bring us to that question.
Okay, thank you.
You're welcome.
Yes.
Well, first of all, congratulations for this amazing session. So I have another question to Sylvia. So if I understood well, so some of those parasites that are circulating during the wet season, they are able to survive. But however, there are some old papers always suggesting that there is no real bottleneck in terms of the whole population of parasites. They don't lose diversity from one season to the next season. So how do you think the parasites can protect their diversity during this cycle through the wet season, dry season?
Thank you. I think it's a lovely question. I think that is at the population level, you don't lose diversity, but because mosquitoes reshuffle the parasites and bring them to non-immune people where they can still grow. So we see the proportions of the different genotypes that we see in the wet season and the dry season in consecutive years is the same. Whatever is broadly present in the wet is going to be also the most represented in the dry. And then you just have to find the non-immune people to keep them going.
Hi, I have a couple of questions. One of them is for Sylvia. So if I understand correctly from your protocol, you treat the patients who are symptomatic, and so the only parasites that you're actually capturing are the ones that have caused asymptomatic infections. And so you have sort of a survival bias there where I wonder if there's actually something fundamentally different between the parasites that are causing these asymptomatic infections that are not going to be treated, and some of these are the ones that are going to be maintained throughout the dry season.
You mean genetically?
Yeah.
Yeah. So we've looked at that. We've never done whole genome sequences. We've looked at a couple of genes, AMA1, MSP2. We never found any biases. I also think that it would be a lot of energy to, if only parasites that are unable to cause disease would survive the dry season, you know that next year severity and disease and pathology is coming. They would have to then generate again all the genotypes that are able to do that. So in our way of seeing things, that it's not a parsimonious enough system. It would be more energetically saving if you have parasites that are able to do both, and they just shift what they can do. Having said that, we have not done whole genome, but everything that we've looked, there's no biases. Even in the ones that get cleared or persist, we don't see differences in genotypes between the two groups. And although we do treat everyone that comes with clinical malaria, we also have plenty of natural clearance without. So it's not as biased as you are putting it, I believe.
Yeah, we can talk about this later. So I also have a question for Professor Saloom. If I remember correctly, there's very different chemistries in some of these rivers. I remember the Rio Negro is very acidic. There's almost no mosquitoes feeding into the Amazon. And then, for example, Rio Madeira, coming from the south, is really muito barrento, very silty. and then they mix around Manaus, and then downstream from Manaus, the river is yet again going to be very different. And I'm just wondering how this affects the mosquitoes, the species, some of the inversion genotypes that you see in different areas, whether there's some adaptation to fundamentally different riverine systems.
So we didn't have enough samples to include from the collection along the Negro River. This is but on just one Barcelos. So, but the main barrier is not the Amazonas River, but the watershed of the rivers. So that is so likely Rio Negro can be a barrier. Yep. But for example, we find mosquitoes not very close from Rio Negro. We don't find along in the water, but we find in another locations along the river. But it's a nice barrier, probably. I don't know. I cannot say more than this. Thank you.
So thank you all the speakers, and thank you for all. I think now is the break, right? Thank you.